Hi, everyone, and welcome to Chapter 11 on evolution and its processes. Evolution is a very big topic, and so, kind of like what we did with Chapter 2, we're going to divide this chapter up into two parts. So, this week, in the first part, we're going to look at evidence of evolution, and then next week, in the second part, we'll look at how evolution actually takes place. So, in this first part, I like to start our discussion of evidence of evolution by showing everyone this photo and asking you to take a moment and try to figure out what it is. And I'll give you a hint by saying that this is a picture of a fossil of a part of a living organism. So some students tell me that they think it looks like leaves or even little pieces of seaweed, but in fact these are feathers, but they're not just feathers. These are feathers from a dinosaur. They come from a fossil of a dinosaur's tail that became trapped in amber almost 100 million years ago. And amber is fossilized tree sap. And you can see in this picture that the tail was very well preserved. And you may ask, how do we know it's a dinosaur and not just a bird? And the answer is that when paleontologists looked at the anatomy of the tail, they found that the vertebrae were not fused together inside of the tail, which they would be if it belonged to a bird. So it's definitely a dinosaur. And when this was discovered, it was one of the first pieces of evidence where you could clearly visualize with your own eyes that there is a real evolutionary link between ancient dinosaurs and modern birds, that modern birds evolved from ancient dinosaurs. Now, that may seem like a crazy thing to say, but science accepts this as factual reality, and not just that modern birds came from ancient dinosaurs, but the entire concept of evolution in general. Evolution is the fact that populations of organisms change over time, becoming genetically different from their ancestors. And there are a lot of misconceptions about evolution. So the goal of this lecture is to lay out the evidence as it pertains to evolution and show you how evolution helps us understand and explain so many important observations about the world of living organisms. And yes, evolution is a theory, but remember from back in chapter 1 that in science, the way that the word theory is used is different from how we use it in everyday language. A theory is something that has proven true across many years by many researchers and countless studies and is considered to be the most reliable level of knowledge that exists in science. So evolution belongs in that camp, and I hope to show you why that is a deserved designation in this lecture. In fact, there's a famous quote about evolution from the Ukrainian-American geneticist Theodosius Dobzhansky, which really sums up a lot of what you'll see in the first part of this lecture, and that is, nothing makes sense in biology except in the light of evolution. What he means by this is that when you really look at some of the features of living organisms, there's so much that really doesn't make a lick of sense unless evolution is the explanation for how it got there. But before we get to those things, I want to start somewhere familiar and close to home. Because the truth is that evolution is something that we can see happening with our own eyes. And one great example is domesticated animals. So let me introduce you to Shrek the sheep. Shrek was a merino sheep, which is the type that produces merino wool, who lived in New Zealand in the 1990s and early 2000s. And you may notice in this photo that Shrek does not look like a normal sheep, and that's because for 6 years, every time the ranchers came around to gather up the flock and shore them for their wool, Shrek successfully duped his owners by hiding in some nearby caves. When he was finally caught, he had enough wool on him to make 20 large men's suits. So, of course, he was shorn and he became sort of a national icon. He even got to meet the Prime Minister of New Zealand. But the point of this story is not to teach you about a celebrity sheep. It's to teach you about a case where we can see how evolution has manifested. Because do you think Shrek's ancestors looked like this? A giant puffball carrying the weight of 20 men's suits and probably can't even see out of its own eyes? If Shrek's ancestors looked like this, they would not have lasted long in the wild. They would have been toast. They would have been easy prey for a wide variety of predators. On the contrary, the ancestors of modern domesticated sheep looked more like this. So this is a mouflon. Mouflons are a species of wild sheep that currently live in Europe and the Middle East. But based on similarities in their chromosomes, the mouflon is believed to be the closest living wild cousin of domestic sheep. And so the mouflon probably very closely resembles what the wild ancestors of sheep looked like. Now this thing, the mouflon, he's much more well suited for survival in the wild. The mouflon is what wild sheep would look like today if humans hadn't intervened. So, how did we get from this majestic, athletic creature to this oversized puffball? And the answer is evolution. In this case, it's humans who are responsible. Archaeological evidence from human settlements show that humans started domesticating an animal very much like the mouflon in Asia between 11,000 and 8,000 BC. And at the time, they were raising these mouflon-like creatures for their meat and their milk and their hide, but wool garments start to show up in the archaeological record around 6000 BC, which tells us that during this window of time of several thousand years, humans were selectively breeding their livestock. Selective breeding is something that humans have done with all kinds of plants and animals, and we're still doing it today. It's based on the principle that people have always known that offspring tend to look like their parents, even if they didn't understand why or how inheritance worked until Gregor Mendel came along. So the basic idea is that you would wait for a sheep to be born among your flock that has a random mutation that causes extra woolliness. Then you would breed that sheep to ensure that its trait gets passed down, and you make more sheep that are extra woolly. And then you keep waiting and breeding that lineage, and maybe the great-great-great-grandchild of the original sheep has another random mutation on top of the original one that makes it even more woolly. So you take that sheep and you breed it to ensure that the trait gets passed down. And over thousands of years, you keep doing this, and eventually you end up something that looks like Shrek, a creature that makes so much wool that it's literally a hazard to its own well-being. It's entirely dependent on humans for regularly scheduled shoring and survival. Shrek is the result of evolution carried out at the hands of humans. So hopefully this gives you an idea of how we can see evolution taking place. We can see how populations of organisms change over time, becoming genetically different from their ancestors. And we don't just see this happening in our friendly domesticated animals. Another place where we can see evolution happening is in our enemies, infectious microorganisms. So this graph shows you how rates of antibiotic resistance among harmful bacteria have been increasing over time. The lines that you see on the graph represent some of the different major classes of antibiotic drugs that are used to treat infections. And as you can see, the percentages of bacteria that are resistant to these drugs is going up. That's because bacteria are developing genetic adaptations that allow them to survive and avoid being killed by antibiotics. The genetic profile of these bacteria is changing over the course of generations because of human antibiotic use. And that's evolution in action too. We can even see evolution occurring, believe it or not, in some human populations. One example is the Old Amish Order in Lancaster, Pennsylvania. This is a community of about 30,000 people who practice an Amish lifestyle, but the group started out much, much smaller. So in the early 1700s, this group consisted of just 20 to 40 families, and one of the founding members of the community carried a rare mutation in his DNA that causes what is now known as Ellis-Van Creveld syndrome, or EVC. People with EVC have 6 fingers on each hand instead of 5, as well as short stature and short limbs. Normally, EVC affects about 1 in 200,000 people across the world. However, because the Amish community is highly endogamous, which is the word meaning that they only marry within the community. And because the community is so small, the rate of EVC is very high because the gene has been spread widely within their small population. So consequently, 1 in 200 individuals in the Old Amish order community have EVC versus 1 in 200,000 in the rest of the world. So the genetic profile of this community has changed over the generations so that it's 1,000 times more common and more likely to have 6 fingers there than it is in the rest of the world. So this is just a small microcosm of evolution in humans. Before we go on, I would like to tap into your expertise and ask you in this first checkpoint to think of another example of evolution that you know of or that you've observed the results of. And as you think on this to find an example, keep in mind that evolution is something that happens to populations over many generations of time. So we've talked about a few examples of evolution to ease us into this topic, but we haven't really dug deep into the evidence. Evolution draws on a massive body of evidence that comes from many different areas of science. So we're going to take a tour of the main areas that supply evidence of evolution. The first of these is paleontology. You may already know that paleontology is the field that examines the fossil record and how it changes and progresses across geological time. The next area is biogeography. Biogeography examines the distribution of living and extinct species across the globe. Then there's anatomy. Anatomy examines physical traits across organisms and species, assessing their similarity and difference. Developmental biology examines the processes of growth and development in different species, especially during their embryonic stage. And finally, molecular biology examines similarities and differences in the composition of organisms and species, DNA and proteins. So in this lecture, we're going to walk through these 5 areas and pull out some highlights from each area, starting with paleontology. So paleontology studies fossils. Fossils are the remnants of living organisms that have been preserved in the form of rocks. Most fossils form when an organism dies and then quickly becomes buried in sediment, like, for example, ash from a volcanic eruption or mud from a mudslide. And then over time, layers of sediment build up and are compressed under the weight of the layers that form above them. And when those layers contain the body of an organism, that combination of the time and the pressure leads the body to become mineralized as rock. And that's what you observe as a fossil. And it's important to note that most organisms never become fossils because their bodies end up being eaten by other animals or they decompose before they can become trapped in a layer of sediment. So for that reason, the picture that the fossil record gives us of the past is by no means complete, but it still reveals some very important insights, as we're going to see in just a moment. But first, let's take a look at some of the different types of fossils that we see. So you can see that fossils come in many different forms. In the upper left is a cross-section of petrified wood, and then down below that there's a dinosaur skeleton. We can see the dinosaur tail preserved in amber, and then on the right-hand side is an image of a couple of sets of footprints that belong to an ancient ancestor of modern humans in Tanzania. And when studying fossils, one of the most important pieces of information to know about them is how old they are. So, how can we tell how old a fossil is? Fossil dating methods fall into 2 categories. The first is relative dating. Relative dating doesn't give you a definitive age for the fossil, but it tells you how old it is relative compared to the fossils around it. So this method is based on the fact that layers of sediment run from the oldest being the deepest layers, and then the most recent ones are the most shallow layers. So the deeper you go, the older the fossil will be. But oftentimes relative dating isn't good enough. Often you need to know an actual number of how many years ago the fossil was formed, because the shifting of tectonic plates can sometimes bring deeper layers to the surface, and so relative dating might not give you an accurate picture. So that's where absolute dating comes in. There are a few approaches to absolute dating, but the most common one involves determining the age of the fossil by measuring the amount of radioactive elements remaining in the sediment. And this technique is called radiometric dating. Radiometric dating is a form of absolute dating that estimates the age of the fossil by studying the decay of radioactive isotopes in the fossil material. So hopefully that word isotope is not completely foreign to you. We learned about it back in chapter 2. So I'd like us to remind ourselves of what it is in a checkpoint. Think back to chapter 2, part 1. What is an isotope? So how does radiometric dating work? Well, it's based on the fact that there's a certain amount of radioactive isotopes of elements that occur naturally in the environment. One example is carbon. Carbon is an element that has two stable isotopes, carbon-12 and carbon-13, which I've highlighted here in the red box. There is, however, a third isotope of carbon on Earth, carbon-14, that is radioactive and unstable. And because it is unstable, over time it naturally undergoes a process called decay, where it gets transformed into a different stable element, which in this case is nitrogen-14. So radioactive carbon naturally decays into stable nitrogen over time. And this happens at a known, well-documented speed. If you have a sample of radioactive carbon-14, it takes 5,730 years for exactly half of that carbon to turn into nitrogen. So by measuring how much carbon-14 is left versus how much nitrogen-14 has been produced, you can get an idea of how old the fossil is. So this graph gives you a sense of how this works. On the left-hand side, the bar that is all blue represents a fossil, or really a dead organism that is 0 years old. So essentially a living organism that has just died and just been buried. It should have a ratio that's pretty much 100% carbon-14 and no nitrogen-14, because there hasn't been an opportunity for decay to occur yet. The bar just to the right of that represents a point in the decay process where there are equal parts carbon-14 and nitrogen-14. It's 50-50. If you find this ratio, it would mean that the sediment layer is 5,730 years old. If you find a ratio of 75% nitrogen-14 to 25% carbon-14, you know that double that time has passed, 11,460 years, and so on. The less carbon-14, the more nitrogen-14, the further you are in the decay process and the older the material is. Now, unfortunately, when it comes to radioactive carbon, this technique is only accurate going back about 70,000 years, because eventually the amount of radioactive carbon gets too small to measure. However, there are other radioactive elements that work the exact same way, but they are accurate going back much, much further. For example, radioactive potassium to argon decay is accurate for dates ranging between 1,000 and 1 billion years ago. And radioactive uranium to lead decay is accurate between 10,000 and 1 billion years ago. So this gives you an idea of how we can tell how old fossils are when we're looking at the fossil record. And when we're looking at the fossil record and dating these fossils, what we see is that patterns of traits follow the progression of time. And I'll give you an example. In 2007, paleontologists in India discovered fossils of a creature they named Indohyus, which literally means India's pig. It was a small, hoofed land mammal that resembled a small deer. However, it had several characteristics that were not deer-like. It had dense, heavy limbs, similar to a hippo, which suggests that it waded in water and the density of its limbs helped to weigh it down when it was wading. It also had a thickened middle ear bone called an involucrum, and that thing has only been seen in the anatomy of one other type of animal, whales. And whales have these special ear bones because it's thought that they play a role in their ability to hear sound underwater. These fossils of Indohyus dated to 54 million years ago. Now, this finding married well with another discovery that had been made some 15 years ago, back in 1992, of an organism that was named Ambulocetus, which literally means walking whale. These fossils dated to 52 million years ago, so slightly more recent in time than Indohyus. And Ambulocetus had a middle ear bone that looked a lot like the middle ear bone of Indohyus. But Ambulocetus also had other adaptations that would have allowed it to hear well underwater, like this pocket of fat in its lower jaw that would capture and transmit sound waves to the ear. And on top of that, the hind legs and pelvis of Ambulocetus, they were structured in a way that would have made it difficult to support their weight on land. So unlike Indohyus, they were probably mostly, if not fully, aquatic. Fossils of another species called Remingtonocetus, which means Remington's whale, had been found also in Pakistan and India, dating between 49 and 43 million years ago. So that's even more recent than Ambulocetus. And Remingtonocetus was missing an important inner ear structure that mammals use to maintain their balance on land. And they also had adaptations in their pelvis and hind legs that would have allowed for efficient swimming. So more evidence that we're moving in an aquatic direction. And then finally, fossils of two species called Basilosaurus and also Dorudon, which is not pictured here, have been found throughout the world, and they date to 41 to 34 million years ago, which is the most recent of all. So in these species, their hind limbs had shrunk down to this tiny, non-functional form that's more along the lines of modern whales. And on top of that, their front limbs took the form of flippers, and their tail was shaped like the tail of a modern whale. So what do we see here? We see a progression that occurs over time, where we start with a hoofed land mammal, and over the course of millions of years, its descendants gradually develop adaptations that are suited to an aquatic lifestyle, while also losing adaptations that are more suited to a land-based lifestyle, until eventually we transition into something that looks like a modern whale. And this picture is, of course, incomplete, but there's a logic here, right? We can see a pattern. There's a direction where transitions have been made from one piece of evidence to the next. So this is what we mean by patterns of traits follow the progression of time. I'll give you another example. Another lineage that can be tracked through the fossil record is the one of ancient equine species, which are the ancestors of modern horses. And without going into nearly as much detail as we did in our last example, over the past 55 million years, the fossil record shows that ancestors of modern horses have evolved in two ways: size and number of toes. So, although modern horses, which are seen on the far right-hand side of this illustration, although modern horses we know have a single hoof, their ancestors possessed multiple separate toes. The fossil record clearly shows a gradual reduction in the number of toes among these ancestral equine species until eventually all of the toes fuse up into a single hoof. And at the same time, equine species became much larger as their habitat transitioned from the forest to the grassland, where they needed to move faster and cover more ground to escape predators. So the bottom line here, both in this example and in the last one that we talked about, is that the fossil record shows us that organisms' traits adhere to a progression that makes sense over time, and that clearly indicates that more recent organisms are descended from more distant organisms, all the way up to modern species. What we don't see in the fossil record is something like this. There's no order here, right? You can't make sense of this progression. If 55 million years ago there were 4 toes and then there was a single hoof and then back to 3 toes, that doesn't support the idea of evolution. But that's not what we see. Another thing we derive from the fossil record is fossils that show linkages between major groups of organisms that we might view as being unrelated. Fossils that serve this role are called transitional fossils. Transitional fossils exhibit traits that are common to both an ancestor and its descendants. And in this sense, they form evolutionary linkages. I'll give you a couple examples to see what I mean. Archaeopteryx is a transitional fossil that dates to the late Jurassic period, about 150 million years ago. And Archaeopteryx had a lot in common with both ancient dinosaurs and modern birds. Archaeopteryx was about the size of a raven, and as you can see from the photo of the fossil on the left, it had wings and is presumed to have been capable of flying. It also had feathers, but in addition to those bird-like feathers, it had a jaw with sharp teeth in it, which birds don't have, and a long bony tail. So it didn't just have tail feathers, it had an actual proper tail. And it also had 3 fingers with claws. And these features are more like a dinosaur. So Archaeopteryx represents one point in time connecting the evolutionary lineage between dinosaurs and birds. Another transitional fossil is Tiktaalik rosier. This creature forms a link between aquatic fish and land-dwelling amphibians. So fossils of Tiktaalik date to about 375 million years ago, and those fossils show some fish-like features like scales and gills, but they also show some features that would be more similar to amphibians, like salamanders, for example. So Tiktaalik had a flattened head with eyes that were on the top of the head instead of being on the sides like fish have. And it also had these bony front fins that would have been capable of bearing weight. And its front limbs had basic wrist bones and finger-like projections. So Tiktaalik is presumed to represent a stage where this lineage was transitioning out of the water and onto the land. Of course, these are just snapshots of the past. The fossil record is not complete because, again, the formation of a fossil requires a very specific set of circumstances where the body of the organism fails to be consumed or to decompose. So it won't ever be possible to identify transitional fossils representing every species that ever existed, but the many transitional fossils that have been uncovered provide us with extremely valuable insight. So now to a checkpoint. As we said, Tiktaalik fossils date to about 375 million years ago. Out of the 3 types of radiometric dating that we mentioned, which could be used to determine the date of Tiktaalik fossils? So that's where we're going to leave things for our brief look at paleontology. Now let's move on to the next area of biology where we see evidence of evolution, which is biogeography. Biogeography examines the distribution of living and extinct species across the globe, meaning where they are physically located. And this is relevant because if there really is an evolutionary relationship between ancient organisms and more recent ones, then we would expect to see most related organisms living in the same geographic areas. And that is what we see, but the matter is complicated by how the continents and land masses of the Earth have changed over time. Because hundreds of millions of years ago, the continents looked a whole lot different than they do today. In fact, one big supercontinent called Pangaea existed from 335 to 175 million years ago. This image here shows the current countries of the world based on where they would have been located in the supercontinent Pangaea. But over time, tectonic plates in the Earth's crust slowly caused the breakup of Pangaea by migrating away from each other, leading to the 7 separate continents that we have today. So this animation shows a model of what geological evidence indicates that breakup process would have looked like. Now here's where it gets interesting. Species of organisms that existed in the age of Pangaea can be found on multiple continents because these fossilized remains migrated with the plates when they moved away from each other. In fact, the habitat ranges of different organisms can show us how the continents used to be pieced together. For example, there's an ancient fern species called Glossopteris, which in this image is shown in the green stripe. These fern fossils can be seen across all of the continents of the southern hemisphere, including Antarctica, where, of course, it would not be able to survive today because ferns don't grow in frozen tundra. There's also an early reptile species called Mesosaurus, which is shown in blue, whose fossils are only present at the southern tips of Africa and South America. And it so happens that the tips of Africa and South America were directly adjacent to each other when they were squished together in the supercontinent Pangaea. However, if we look at fossils that date to after the breakup of Pangaea, after 175 million years ago, when the continents were already separated, we see that similar species tend to be clustered together in their current locations rather than spread across different continents. For example, those fossils that we talked about that are the ancestors of modern whales, all of those date to periods of time long after the breakup of Pangaea, and all of those fossils were found in Pakistan and India, which, if geography is not your thing, Pakistan and India, they directly border each other, so same general area. Another example, 70% of existing marsupials, which are mammals with pouches, are found in Australia and only in Australia. Fossils of the earliest ancestors of humans are all found in Africa, most of them in southeastern Africa specifically. And out of 2,000 species of fruit flies in the world, a full 25% of them live only in Hawaii. So these are just a few examples that show you how more recent species are clustered because they are related and they're descended from more ancient species that have habitat ranges that were consistent with Pangaea. So that wraps up our look at biogeography. Let's move on next to talking about anatomy and developmental biology. And we're going to talk about these together because they're so closely related. Anatomy is the examination of physical traits across organisms and species, and developmental biology examines the processes of growth and development, especially during the embryonic stage. One piece of evidence of evolution that we see when we look at the physical traits of organisms is called homologous structures. Homologous structures are body parts that appear similar in separate organisms because they evolved from a common ancestor. For example, vertebrates, meaning animals with a backbone, they all have the same number and arrangement of bones in their limbs, even though those limbs serve very different functions. So here we see the bones in the arm of a human, the leg of a dog, the wing of a bird, and the front flipper of a whale. You can see that the bones, the ones that are homologous to each other, they've been colorized in the same way. So the red bones are the same as the red bones, the brown bones are the same as the brown bones. And you can see that the construction is basically the same. And that's true, even though a dog uses its leg to walk, a bird uses its wing to fly, a whale uses its flipper to swim, humans use their arms for all sorts of things. Even though they perform different functions, the bones are all put together in the same way. And the reason why this is the case is because all of these vertebrate animals, they came from a common ancestor that passed down basic instructions in the DNA for how to make these limb bones, and they just became adapted to suit various different purposes over millions of years. Another example of homologous structures is in the vertebrae, which are the neck bones seen in the necks of animals. So nearly all mammals have exactly 7 vertebrae in their neck. The 3 examples that you can see here are of a mouse on the left, a giraffe in the middle, and a human on the right. Each of them has 7 vertebrae, even though the mouse's vertebrae are tiny and the giraffe's vertebrae are enormous. You would think it would make more sense for the giraffe to have a larger number of vertebrae to suit its longer neck, but no, it still has 7 because the DNA in the giraffe spells out a master plan that involves having 7 vertebrae. And that master plan was passed down from an ancestor that is shared with humans and mice. We also see homologous structures in plants. For example, among plants that make flowers, which not all plants make flowers, we'll get to that a few chapters from now, but among the ones that do, they all have the same flower parts, some of which we talked about in our chapter on inheritance. There's an anther that carries the pollen, there's an ovary that carries the ovules, and this is seen across a huge variety of different flowers that may look superficially to be very different from each other, but their fundamental construction is the same. Now, it's important not to confuse homologous structures with analogous structures. Analogous structures are body parts in different organisms that evolved independently to serve the same purpose. So, for example, bird wings and insect wings. Both of these serve the same purpose, but their structure is fundamentally different. For example, bird wings contain bones, insect wings do not. So cases like this are not evidence of common evolutionary ancestry. Instead, they are the result of convergent evolution, which is where separate lineages evolve an adaptation that converges on the same appearance and performs the same function. There are also certain types of homologous structures that are called vestigial structures. Vestigial structures are unused structures that have no function for an organism, but they're clearly homologous to structures found in other organisms. For example, some species of birds have wings, but they can't fly. Why have wings then? Why not have arms or something more useful? It's because they didn't have a choice. Their ancestors passed down the genes for wings, so that's what they got, even though those wings are obsolete now. Another example, there's a species of fish called blind cavefish, and blind cavefish live in pitch black underwater caves. They therefore have no need for eyes. In fact, they don't have eyes at all, but they still have eye sockets. Why have an eye socket but no eye to go in it? Again, they didn't have a choice. If they got the genes for eye sockets, they're going to make eye sockets, and that's what's passed down from their ancestors, even if there's no eyes to go in those eye sockets. Here's a really wild one. Snakes have pelvic bones, tiny little bones that make up a pelvis that you can't see from the outside, but they have no hind limbs to attach to them. Why would a snake need a pelvis? It doesn't. The only way this makes sense is if the pelvic bones are evolutionary leftovers that got passed down from the ancestor of the snake, which did have 4 limbs and therefore needed a pelvis. Getting back to an example that connects to our paleontology discussion, whales have internal hind limb bones, but no hind limbs. So you can't see these bones from the outside, but if you were to dissect a whale, you would find that they have these tiny, non-functional bones for hind legs, which is an artifact of their evolution from a land-dwelling mammal, as we saw in the fossil record. And even humans have vestigial structures. Humans have tailbones, even though we don't have tails. We have ear muscles, even though we can't move our ears in a meaningful way. We have an appendix, which is a superfluous organ that does nothing for us aside from occasionally rupture and require surgical removal. So why do we have these things? Because we're descended from an ancestral organism that did have a tail and could move its ears, and for whom the appendix actually did something beneficial. So now let's pause for a few checkpoints to test our understanding of these concepts. The hearts of mammals and reptiles have a similar construction that was passed down from a common ancestor. This means they are an example of a what structure. Next, hoofed mammals have dew claws that are homologous to dew claws in other mammals, like the dog that you see on the left here. The one claw that's up on the side of the ankle is called a dew claw. And hoofed mammals have them too, but they serve no function. This makes them an example of a what structure. And then third, many bacteria have a structure called a flagellum that allows them to move throughout their environment. Human sperm cells also have a flagellum that allows them to move. However, the two types of flagella are constructed differently, they move in different ways, and they're produced by entirely different genes. They evolved independently. This makes bacterial and sperm flagella examples of what type of structure? So now let's talk about evidence of evolution that shows up in embryos. An interesting exercise that I like to have students do on this topic is to look at the 5 embryos that you see lined up here. So each of these 5 embryos is a vertebrate, meaning they all belong to that category of animals with backbones. One of them is a human. Can you guess which one is the human? Take a moment and try to figure it out. And this is a really hard exercise because they all look so similar. But it turns out the very first one is the human. Second one is a mouse. The third one is a bat. Fourth one is a chicken. And the last one is an alligator. So we've got a wide variety of organisms. But despite this, they look pretty similar. Each of them has 4 limb buds, a tail, segmentation along the back, gill arches, and much more in common. The period of embryonic development is a hot spot in time for homologous structures and for vestigial structures. It is common to see vestigial structures appear during embryonic development and then later disappear. For example, embryonic humans have tails, full tails, and gill arches. Embryonic horses have 5 toes. Embryonic whales and dolphins have hair. Now, over the course of gestational development, these features go away, so they're not present by the time the organism is born. So given that they'll eventually go away, why do these things appear when these creatures are embryos? It's because of homeotic genes. Homeotic genes dictate major body parts and systems early in the organism's development. They basically dictate the master plan for the body, meaning major features like limbs, organs, organ systems. And these homeotic genes tend not to change a lot. They mutate very slowly because changes in them can be catastrophic. If something goes wrong with the gene that says you should have legs or lungs, those are major problems. So these genes tend not to change a lot. And because they tend not to change a lot, they are shared widely across different groups of organisms. In this illustration here, you can see a series of color-coded homeotic genes and which part of the human body they dictate the development of. And these are very similar to the genes found in other animals like mice, which can control their master body plan as well. Even in organisms as different from us as insects, you find similar genes that outline the development of the master plan for the body. However, even though homeotic genes are active early in development, when an organism is still an embryo, they can be overridden by the activity of other genes later in development. So this is evident from the fact that in human embryos, the tail disappears by 8 weeks. In horses, the embryos have 5 toes that eventually fuse to become 1 hoof. And in whales and dolphins, the hair falls out and it doesn't grow back. So to summarize, homeotic genes control early development, and they cause diverse ranges of organisms to develop similar traits early on. But then later in development, other genes can come into the picture and override the action of the homeotic genes, which is a less risky route than making big changes to the homeotic genes themselves because they're so important. However, and this is key, the genes for these traits are still in the DNA. Humans still have the genes for a tail. Horses still have the genes for multiple toes. And sometimes the override mechanism that stamps these things out can fail. When this happens, the result is called atavism. Atavism is the reemergence of a trait from an ancestral organism. It comes about when mutations in the override mechanisms cause an organism to revert back to the traits that were passed down from their ancestor. And again, I'll give you a few examples. Humans can be born with tails. This is extremely rare, but sometimes that embryonic tail doesn't go away, and that's just what you see on the left. Whales and dolphins can be born with hind flippers, instead of just having tiny invisible hind limb bones hidden on the inside. That's what you see on the right. Horses can have extra toes that failed to fuse with their main hoof, such as the case seen on the left. Snakes can be born with little non-functional legs, like what you see on the right. Truly bizarre, right? But what explains this weirdness is the fact that the genes for their master plans are all the same. They're all plays on the same basic construction. They've just diverged into different forms over time. We'll stop here for another checkpoint. Occasionally, humans are born with more than 2 nipples, which is a condition known as supernumerary nipples. This is an example of atavism. Think about what this means. Why would this be an example of atavism? What does it say about the ancestors of humans? So we're now moving on to our last area of exploration of evidence of evolution, and that is molecular biology. This is also the newest and most recent field in terms of technology. Whereas the other four fields have been studied for hundreds of years, the ability to determine the sequence of an organism's DNA and proteins has only existed on the scale of decades. However, despite being the most recent, molecular biology is the most powerful and definitive tool for establishing relationships between organisms. Because whereas some of these other fields may provide us with evidence that can be hard to interpret in certain cases, DNA evidence clearly proves the degree of relatedness between species, just like it proves the degree of relatedness between individuals. So we all know that the more closely two individuals are related to each other, the more DNA they share. So for example, you share more DNA with your siblings than you do with your cousins, because your closest ancestor that you share with your siblings is your parents, but the closest ancestor that you share with your cousins is your grandparents. So the farther back you have to look to find a common ancestor between you and any other person, the less similar your DNA is. This principle is also true across species, not just individual people, but the entire tree of life. So when we look at the DNA of various organisms, we can see that the percent similarity gets lower the farther away you get in the tree of life. This table here shows you the percentage of DNA that's identical to humans in each of these organisms. 96% of the DNA in chimpanzees is identical to that of humans. We're only 4% different. In mice, 88% of their DNA is identical to humans. In zebrafish, 73%. Fruit flies, 47%. Even rice, a plant, 25% of its DNA is still identical to humans. Now, the fact that chimpanzees share 96% of their DNA with humans, does this mean that chimpanzees are the ancestors of humans? No, it doesn't. This is one of the most common misconceptions about evolution. The idea of evolution does not claim that humans evolved from chimpanzees. That would be like saying you evolved from your cousin. Humans and chimpanzees are very, 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 very distant cousins. But as far as species go, they're closely related. In fact, out of all the species in the world, chimpanzees are our closest cousins according to the DNA evidence. Next closest are gorillas and orangutans and various types of monkeys. And as you get steps further away, you have to go back further in deep history to find a common ancestor. Now, as you know from our last chapter, the genes in DNA provide the instructions for making proteins. So, as a result, we see the same trend in proteins, where the more closely related organisms have more similar looking proteins. Of course, to see this trend, you have to look at a protein that is found in a wide diversity of organisms. One example of these is the cytochrome C oxidase protein. This is a protein that's involved in cellular respiration. And you may remember from chapter 4 that cellular respiration is the process that cells use to produce energy in the form of ATP. It's the series of chemical reactions that break down sugars and turn their energy into ATP. And cytochrome C oxidase is one of the proteins embedded in the inner membrane of the mitochondria through which hydrogen ions are pumped during the very last stage of the process. I've circled that protein in the illustration. So because all cells have to produce energy, virtually all eukaryotic cells have this protein in their mitochondria. Plants, animals, fungus, they all have it. So that makes cytochrome C oxidase a good protein to compare across different organisms to study how related they are. And this table here shows you how similar the cytochrome C oxidase protein is in a variety of different organisms compared to humans. The protein is 105 amino acids long, and in chimpanzees, all 105 of those amino acids are identical to the ones seen in humans. In rhesus monkeys, all but one of them are identical. In horses, 12 out of the 105 are different, In tuna fish, 22 are different. And in thale cress, which is a type of mustard green plant, 39 of the amino acids are different. So as you can see, with proteins, there's the same trend that we see in DNA. The closely related organisms have a greater degree of similarity in their proteins. As you get farther away in the tree of life, the similarity becomes less and less. Now, as I said, molecular biology is the most recent area to start contributing to our understanding of evolution. And when this technology came on the scene, in some ways it really shook things up, because until DNA data was available, scientists were forced to come to conclusions about how related two groups of animals are based on their physical characteristics alone. So, for example, whales and hoofed mammals were classified into two different groups called orders. Cetacea was the name of the group that contained whales, and Artiodactyla was the name of the group that contained hoofed mammals. And when you just look at their anatomy, that makes sense. They look nothing alike. However, when DNA evidence came along, and scientists actually started looking at the DNA of whales and hoofed mammals, the DNA told them that they were actually more closely related than their appearances would lead you to believe. And so since the 90s, the two groups have been merged into one singular group called Cetariodactyla. Another example of a relationship that was confirmed by DNA evidence is the one between dinosaurs and birds. So in 2006, researchers in Montana actually recovered some unfossilized T-Rex bone, meaning it was not just the mineralized mold of a bone, it was actual bone material that had managed to be preserved for over 65 million years. They had this partially fossilized T-Rex leg bone that was so large they couldn't fit it in the helicopter that they were using to transport it. So they broke it in half to make it fit in the helicopter, and when they did, they realized that the inner portion was still bone. It wasn't fossil. So they proceeded to extract several collagen proteins from the bone for study, and they compared the amino acid sequence of those bones to a variety of other organisms. And what they found is that 5 out of the 6 proteins were identical to the collagen proteins in chickens, but not to the collagen protein in amphibians like newts and salamanders. Of course, just looking at a salamander and a chicken and a picture of a dinosaur, you might think that the salamander is more closely related to the dinosaur based on their appearance. But the sequence of the collagen proteins tells a different story. It's actually birds that are the closest living relatives of dinosaurs. One other evolution puzzle piece that molecular biology has revealed is the presence of pseudogenes in organisms' DNA. Pseudogenes are non-functional pieces of DNA that are homologous to functional genes found in other organisms. They're basically the vestigial structures of DNA. They're genes that are broken, but they're still there. An example of this is the gene for L-gulonolactone oxidase. L-gulonolactone oxidase is an enzyme that's involved in the production of vitamin C. And that's what you can see on the slide here. This is the molecular structure of vitamin C. The gene for this vitamin C producing enzyme is found in nearly all mammals. However, the version of the gene that's found in primates, including humans, has catastrophic mutations in it. The gene is still there, but the mutations in the gene make it non-functional. This is why humans need to get vitamin C as an essential part of our diets. We don't have this enzyme, and if we had it, our bodies would be able to make vitamin C on their own steam. So, if this gene is broken, why is it still there in our DNA? Doesn't make sense for it to be there unless you look at this in the broader context of evolution and understand how it was passed down from a mammalian ancestor where it did work properly, but it got mutated somewhere along the way, and now we walk around with this broken relic of a gene instead. So we just covered a lot of ground in a short amount of time. I think a recap is in order. Here is a summary of the evolution evidence that we've talked about. Within the fossil record, patterns of traits follow the progression of time. There's a logic and order to the fossil record where you can see gradual transitions happening across long periods of time. Some of the fossils that we see are transitional fossils, and they have traits that are shared by ancestral groups of organisms as well as descendant groups of organisms, and they show how different groups are linked to each other. The geographic distribution of very old fossils matches what you would have expected to see when all of the continents were merged in the form of Pangaea. But if you look at more recent organisms that existed after Pangaea broke apart, you see that related organisms tend to be clustered together. If you look at organisms' anatomy, you find homologous structures across related groups of organisms, as well as vestigial structures that lack a function at all, and these are especially common during embryonic development. Related organisms tend to look extremely similar during embryonic development because they have very similar homeotic genes that control their master plan. But cases of atavism can show the reemergence of ancestral traits that are still hidden in the DNA. And then finally, DNA and protein sequence data from molecular biology confirms the relationships of organisms that we see in all these other fields of study. And what explains all of this evidence put together is evolution, that populations of organisms change over time, becoming genetically different from their ancestors. All of this evidence makes sense once you understand that all life is related, younger species are descendant from older species, organisms accumulate changes and diverge from each other over long periods of time, and the organisms that exist today are all cousins, quote unquote, with varying degrees of relatedness depending upon when they diverged. So this wraps up our discussion of evidence of evolution. In our next lecture, we're going to talk about how evolution actually works, but I'm going to leave you with a few checkpoints to round this lecture off. In 1974, researchers excavated a fossil skeleton of a hominid, a human-like primate, in Ethiopia. The skeleton was nicknamed Lucy and given the species name Australopithecus afarensis. The fossil was 3 feet 7 inches tall and an estimated 64 pounds. She was bipedal, meaning that she walked upright on 2 legs, and her humerus, which is the long bone in the upper part of your arm, was 84% as long as the femur, which is the long bone in the upper part of your leg. Now, in chimpanzees and other apes, the upper arm is about the same length as the upper leg. The humerus is 97.8% as long as the femur. But in humans, the figure is 71.8%. Our legs are proportionally larger than our arms. So Lucy had characteristics that made her an intermediate between other apes and humans. This makes Lucy's fossil an example of a what fossil between apes and humans. Next, many animals have a gene called CMAH in their DNA. CMAH encodes an enzyme that is responsible for creating a carbohydrate identity marker that's present on the surface of cells. So remember from back in chapter 3, one of the features of cell membranes is that they have carbohydrates sticking off of the outer surface, which is what's highlighted in the yellow boxes here. And these carbohydrates can behave like the cell's ID badge and serve as identity markers for recognition by other cells. So CMAH is a gene that makes an enzyme that makes one of these carbohydrate ID markers. But in humans, the CMAH gene is broken. It is missing a 92 nucleotide long section of DNA that is seen in other great apes, but not humans. With those 92 nucleotides missing, the enzyme made from the gene is non-functional, and therefore the carbohydrate identity marker is not made. And the absence of this identity marker leaves human cells susceptible to being infected with the most deadly version of the microorganism that causes malaria. In humans, the CMAH gene would be considered a what? And lastly, after taking all of this in, I'd like you to share your thoughts on Dobzhansky's famous quote that nothing makes sense in biology except in light of evolution. What's your take on him saying this after what we've learned here? And once you're finished with this question, you're finished with this lecture.